Welcome to the second video in the DNA replication unit. In this video, we will discuss the various laboratory techniques used to manipulate DNA and their uses in genetic engineering. Scientists are now able to isolate, identify, and sequence DNA fragments by exploiting the structural and physical properties of DNA and the principles of DNA replication. We use these DNA techniques to determine genetic risk factors, identify DNA at crime scenes, create recombinant DNA, and genetically modify organisms, and give insight into how closely related two species are to each other, among many other things. Listed here are some of the key DNA manipulation techniques that are routinely completed in molecular laboratories. The first technique we will discuss is PCR, which stands for polymerase chain reaction. PCR is a method of amplifying or making many copies of a targeted region of DNA. This method was developed based on our understanding of cellular DNA replication and can be thought of as replication in a test tube. There are three steps to PCR. The first is denaturation. Denaturation is a way of artificially completing the role of helicase in DNA replication. By heating double-stranded DNA to 95 degrees Celsius, the hydrogen bonds holding the two strands together will break, separating the strands from one another. The second step is annealing. In this step, short sequence of DNA primers that are part of the PCR reaction bind to the single-stranded DNA as the temperature is cooled. The DNA primers are made in a lab to match the sequence that you are trying to make many copies of. They are taking the place of the RNA primers made by primase during DNA replication. The third step is extension. In this step, DNA polymerase is used to synthesize a new strand of DNA, building from the 3' end of the DNA primer, the same way that cellular DNA polymerase synthesizes new DNA strands in the cell. These three steps make up one cycle of PCR. In order to make many copies of DNA, 30 to 40 cycles of PCR are performed, so after the extension step, the process starts back over at denaturation. In order for PCR to work successfully, scientists use special DNA polymerase called Taq polymerase. This enzyme was isolated from bacteria that live in the hot thermal vents of the ocean. Because the enzyme functions at high temperatures naturally, it is not destroyed when the temperature is increased back to 95 degrees Celsius during the next denaturation cycle. Watch the video on the next slide to see PCR completed. In addition to amplifying DNA, it can also be useful to cut DNA at specific sites. Cutting pieces of DNA allows pieces from the same or different organisms to be brought together in recombinant DNA technology. This is used for generation of genetically modified organisms. Or it can be used to cut large genomes into small pieces for further analysis, such as in crime scene analysis. The method for cutting DNA makes use of a class of enzymes called restriction enzymes that recognize and cleave specific short nucleotide sequences in double-stranded DNA. These sequences are called restriction sites. This table includes a list of various restriction sites recognized by their corresponding restriction enzymes. You should notice that each sequence is a palindrome. A palindrome is a sequence that is spelled the same way forwards as it is backwards. For example, the BAMH1 sequence, seen here, reads GGACT, going 5' to 3' this way. It also reads GGACC, going 5' to 3' that way. The enzyme would then cut the strand of DNA at the location marked by the arrow. This is an example of how the enzyme EchoR1 would cut a piece of DNA into two separate sequences at its GAATTC restriction site. Many enzymes, such as EchoR1, leave sticky ends after cutting DNA. These are short, single-stranded regions of DNA here and here that are useful if you want to attach one fragment of DNA to another. When a restriction enzyme is used to break up a whole genome, such as the bacterial genome here, the specificity of the enzyme ensures that each cell of the organism provides the same set of smaller fragments. In this example, you have a set of fragments containing one piece of each of these sizes listed around the genome. This set of fragments can be used to make a DNA fingerprint unique to that organism. This is similar to what occurs in crime scene or paternity testing. Another key technique in molecular biology is gel electrophoresis, which allows for the separation of molecules based on their size. Gel electrophoresis can be used to confirm your PCR-amplified DNA that you were expecting, or to separate DNA molecules after digestion with a restriction enzyme. Gel electrophoresis separates DNA molecules based on size using a porous material called a gel. You can think of a gel as very dense jello. To begin the process, DNA samples are placed in the small holes called wells at one end of the gel. An electrical current is then passed through the gel. You must make sure the negative end of the current is at the end with the wells, and the positive end is down farther away. This is because DNA is negatively charged due to the phosphate groups in its backbone, so it will move towards the positive charge through the attraction. Small pieces of DNA are able to move through the gel more easily than large fragments, so when you examine the gel, the shorter fragments will be closer to the bottom of the gel. If you load a solution of control fragments with known sizes, called a DNA ladder, then you can use that to determine the size of your fragments. It is also important to note that DNA is not visible to the naked eye, so in order to use the observed separated DNA fragments, you first need to dye your DNA. The three techniques just discussed are key methods used for recombinant DNA technology. Recombinant DNA is a DNA molecule that contains two or more different sources of DNA. The method takes a donor fragment of DNA that contains a key protein encoding gene or any DNA segment of interest, for example, the human gene for insulin, and it inserts that into another piece of DNA referred to as the vector sequence. You can think of vectors as the carriers of the donor DNA. The donor segment could be amplified by PCR or cut from a chromosome using restriction enzymes, as seen in this figure. The vector DNA must also be cut with the same restriction enzyme. This generates two separate pieces of DNA with matching sticky ends. These sticky ends will be able to form complementary base pairs with one another, as A's pair with T and T's pair with A. The two pieces are then joined together to make a piece of recombinant DNA using the enzyme DNA ligase. This is the same enzyme that joins the Okazaki fragments together during DNA replication. Many times, this recombinant DNA can then be inserted into a bacterial cell. The bacterial cell then replicates, making many copies of the recombinant DNA. These bacteria are used in various industries to produce molecules of interest. In this example, the bacteria are producing insulin that we use to treat diabetes. Recombinant DNA technologies are not only used to genetically modify bacteria, but also to genetically modify laboratory organisms, agricultural crops, and domesticated animals. These organisms are called transgenic organisms or genetically modified organisms, abbreviated as GMOs. GMO organisms include sheep that produce human protein in their milk to treat emphysema, salmon with increased growth hormone to get to market sooner, and crops that resist various pests.